Mr. Cameraman, I'm here! Finally. Hey there, folks! I have Chica to tell. I would like to share what happened yesterday because I want to show you all how kind I am. Not that I'm boasting, but just
Here we go again.
Make her stop,
por favor.
Whatever, you invisible creature.
Blah blah blah.
So going back, let's begin with the story. One pleasant day at La Salle's University, a section was having a discussion on biology.
Okay class, now that we are done with our first topic, let's jump to the next one. Cell cycle and cell division. When you hear the word division, what is the first thing that comes to your mind? Anyone?
Suddenly, the bell rang. Ding, ding. The students were happy because finally, it was time to go home.
Seems like we ran out of time. Students, I guess we'll just tackle this tomorrow.
Children started to pack their things. But then, their teacher announced something. Can you guess what is it?
Since we didn't finish the discussion today, please do some research about how animal cell division occurs. You can watch it on YouTube or go to our school lab and write it down in your notebook.
Hearing that, the smiles of all students quickly faded. But not this student. After class, this student goes to the school lab. Earlier, she asked her classmates if any of them would want to go to school lab with her. But no one wanted to. They said that it was boring and time-consuming. They'd rather watch on YouTube or search on Google. So she went alone. Once she arrived, she saw a microscope, which she can meet on the specimen stage. She immediately took a look at it and was shocked to see that there was something looking at her.
Hello there!
Oh my god, what was that?
Suddenly, a voice was heard on the scene. She helped to ease the girl's anxiousness. After that, they started to talk. Calm down. We don't bite over ever. Just relax. Take a deep breath. Okay? Inhale. Exhale. Yeah, that's right.
Who are you and where are you?
Hi, I'm Devin. Well, I'm here in the ceiling. You can't see me. Nobody can. Because I'm invisible. How about you? Who are you? Why did you come here? It's so rare to see a student here in the lab.
I'm Naelea. I'm here to observe how cell cycle and cell division works. Because that's her assignment.
Without second thoughts, Devin, aka me, yes, my name is Devin, decided to offer Naelea a help so that she can answer her assignment. Good thing. I'm in a good mood today. I might be able to help you with that. Take a look at the microscope again.
Hello there.
I told Naelea that the microscope is magical. It has the ability to show the cells and inform you about how the process of cell division works.
Yo, that's cool.
I told Naelea what would happen and what she would do. I even asked help from my friends, which is actually a cell, to show Naelea how it happens so that it's much more understandable, and because I'm a great creature, a good, a beautiful and pure soul,
Time is running.
Hey G1, can you help this kid understand cell division more?
I guess so!
Good. Tell them that they have a kid to teach her.
Yeah, alright.
And you? Aliyah.
It's Naelea.
Whatever, you're gonna shrink, much smaller than dust. You'll be able to interact with the cells, so stay calm. They're gonna teach you everything you should know. Now when you say, magical microscope on the table, make me tiny, so small. You're gonna be so tiny. To get back to your normal size, you should say, magic microscope, I figure out what my hows and whys. Please get me back to my normal size.
Okay, got it. Magic microscope on the table. Make me tiny, so small.
Naelea was stuck inside the microscope. She was as small as a cell. She was amazed about what happened.
Oh my gosh, it worked. Wow. This is amazing! Oh my god.
She saw three faces at first. Devin, aka me, again explained to her that the three faces she's seeing is the interface. After that, I introduced Naelea to the three subphases of interface. This is the cell cycle. It is the life of a cell from the time it is formed during division of a parent cell until its own division into daughter cells. Over there you can see the three parts of the interface. Say hi to G1 or GAP1.
Hello.
Oh, hi.
This is S or DNA synthesis.
Hi there!
Hello.
And G2 or GAP2.
Hi.
Oh, hey. Nice to meet you.
90% of the cycle includes them. After Devin introduced the interface, she told them to help Naelea. Make sure that she learned something. If not, you're gonna regret that you went through the process of growth. G1 then started to tell Naelea what he has to know.
Hi Naelea, I am G1, can be called as GAP1, the first phase under the interface. I am in charge of the growth cells. In this phase, the cell grows physically bigger. It makes proteins and organelles needed for normal function and DNA synthesis. Here proteins and RNAs are synthesized.
G1 then showed Naelea how it happens.
Oh, wow. So, I have a question. How would you know if the cells are ready for the next phase?
G1 walked Naelea to the checkpoint and explained to her what happened there.
Is the G1 checkpoint. If the cell hasn't grown much or if it's not yet ready to undergo the phase, it would be blocked by the checkpoint. Then the cell would exit the cycle and stay in G1.
Another question again. Why does it have to be examined if it is ready for the next phase or not?
Let S or synthetic phase answer that. Hey S, it's your turn buddy.
S phase then took over. She brought Naelea to where the S phase takes place. S phase, together with the other cells, welcome her.
Hi Naelea! Welcome to S-Space!
Oh, thank you. Shall we proceed?
S phase then started to explain Naelea everything she needs to know.
I am the S-Phase, the second phase of the interface. Cells that were approved at G1's checkpoints made by their way to me.
Naelea then asked the question she asked G1 before she left, and S phase answered it.
I asked G1 earlier and He said that you'll be the one that could answer this. I was just gonna ask if um how I mean why do cells have to be examined if they are ready for the S phase or not?
That is a great question. It has to be examined because S-phase is in charge of duplicating the genetic material, such as chromosomes and DNA. If a cell is not fully developed in the first phase, my phase would also duplicate the underdeveloped cells. Once it happens, it can cause abnormality to the organism we're in charge of.
S phase then showed Naelea what happens in his phase and explained it to her.
Wow.
That is what happens in the S phase. Chromosomes and DNA are being duplicated so that there be enough DNA to be split into daughter cells. Naelea then take S phase and proceeded to G2, or what we call GAP2.
Bias phase, thank you.
Naelea begins to enjoy her journey where the cell process took place.
I've never seen a place like this. This is amazing.
While Naelea is still amazed of where she is, someone tried to get her attention.
Woo!
Psst, 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 psst. Naelea was shocked and scared when she heard that sound.
Oh my gosh. What is it? Psst, psst. Oh my gosh. What is it? Hello? Is somebody here?
Over here, Naelea.
Who are... Oh, I've seen you earlier. You're the grumpy one.
As Naelea's excitement arise, G2 started to explain what happened to the cell in his stage.
Hi, I'm Jidu. Wanna know something about my stage?
Of course.
In my stage, I allow cells to grow more, make more protein and organelles. This is where also cells are preparing for actual cell division.
But why do cells need to grow bigger before going to the actual cell division?
Cells need to grow more before the actual cell division because daughter cells need to have the same amount of organelles to contain them all.
Oh, now I get it. Thank you. Thank you. I learned a lot from you.
What a gentleman you are, G2. Now, this is where the exciting part begins. Time to shine, Prophase. Oh, it's my turn now.
Yeah, I think so.
Anyways, hi Naelea, I'm Prophase, the first stage of Mitosis.
Hi, Prophase. It's nice to meet you.
Nice to meet you too, but since our time is only limited, let's get started. Are you ready?
I'm ready.
Without further ado, Prophase explain what is happening in her stage. Okay, in my stage, the nuclear envelope begins to disappear as the centrioles move in opposite direction toward the nucleus. Chromatin fibers condense into distinct chromosomes as they become more tightly wound. The nuclei will subsequently disappear and each duplicated chromosomes will then appear as two sister chromatids that are connected at their centromeres. As the microtubules between the centrosome lengthen, the mitotic spindle will start to develop and the centrosomes will start to migrate apart. Look right there, see? But Naelea is quite confused to that long explanation made by Prophase. So Naelea asks something to Prophase.
Whoa, whoa, whoa. Wait, is there any simple explanation for that?
Of course there is. In short, mitosis begins in prophase and then the DNA condenses and begins to show as chromosomes. The nuclear membrane disappears and mitotic spindles grow out of centrioles.
Wow. Now I get it. Thank you, Prophase.
You're welcome. I hope you have great journey ahead. Get ready to face Pro-Metaphase. Bye-bye! Naelea was brought to the dimension of Pro-Metaphase.
Hello, Naelea.
Hello there.
After greeting each other, Pro-Metaphase stated things about her face.
Did you know that between prophase and metaphase, we have what we call prometaphase?
Naelea shakes her head, an indication that she doesn't know what Pro-Metaphase is talking about. Still, Pro-Metaphase proceeded.
Listen carefully, Naelea, as I tell you what's going on in prometaphase. This is the phase of mitosis that begins when the nuclear envelope starts to break down. The breakdown of the nuclear membrane allows the chromatin structure to condense and thicken into chromatids. After that, the growing microtubules cover the area where the nucleus used to be, so that they can attach to special proteins called kinetochores, which have assembled on the chromosomes at the centromeres. You know what, Naelea? The microtubules that becomes attached to the kinetochores are known as kinetochore microtubules, which jerk the chromosomes back and forth. The kinetochore is important as it joins the sister chromatids at the centromere, while the non-kinetochore microtubules interact with those from the opposite pole of the spindle, lengthening the cell.
Both said goodbye to each other and Naelea continued with her journey.
That's all for now, Naelea. You can proceed to the next phase of mitosis, which is metaphase. Until next time, bye!
Naelea then goes to the next phase, which is metaphase. This phase welcomed her as well. After Metaphase introduced herself, she started to talk, not letting Naelea said a word. Well, Metaphase is kinda impatient, so
Hey!
just kidding.
Hi Naelea. I know you are here now because you are curious about the pace. So we are here to help you to solve your curiosity. By the way, I'm Metaphase. Naelea, the metaphase is the third phase of mitosis, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During metaphase, the cell's chromosomes align themselves in the middle of a cell through a type of cellular tug of war. The chromosomes, which have been replicated and remain joined at a central point, called the centromere, called the sister chromatids. Prior to metaphase, protein formation called kinetochores form around the centromere. Long protein filaments called kinetochore microtubules extend from poles on either end of the cell and attach to the kinetochores. During metaphase, the kinetochores microtubules pull the sister chromatids back and forth until they align along the equator of the cell, called the equatorial plane. There is an important checkpoint in the middle of mitosis, called the Metaphase Checkpoint, during which the cell ensures that it is ready to divide. Once the cell has establishment that all of the chromosomes are properly aligned and that the kinetochores are correctly attached, The cell enters the fourth phase of mitosis, known as anaphase.
Naelea didn't have a chance to ask question because she don't want to interrupt Metaphase. Yet Metaphase explained everything so well that Naelea doesn't have to ask questions. After the brief discussion, Metaphase said bye to Naelea and suddenly vanished, leaving Naelea stunned and alone.
So, you can proceed to anaphase, Naelea. Bye.
Goodbye. What's with her?
Naelea was shocked when she heard someone calling her.
Naelea. Yoo-hoo. Hello. Hi.
Naelea looked back and saw the next face waiting for her.
Oh, hi.
Anaphase introduced herself.
Hi, Naelea. I'm Anaphase, the fourth phase of mitosis. Nice to meet you, Anaphase.
Anaphase starts to tell Naelea things about her. Naelea just carefully listens to Anaphase.
In this phase, the chromosomes move toward to the pole of spindle, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells.
Anaphase continued to explain the process of her stage, while Naelea just agrees to everything she says.
Before cell division, each chromosome duplicates to form two identical parts called sister chromatids, attached to each other at a region called the centromere. During anaphase, the division of the centromere releases two daughter chromosomes, one for each daughter cell.
Naelea was delighted to see how Anaphase process works. She looks really amazed that chromosomes divided. Anaphase still has something to say, so she speaks again.
Naelea, did you know that in anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell? The protein glue that holds the sister chromatids together is broken down, allowing them to separate. Each is now its own chromosome. The chromosomes of each pair are pulled towards opposite ends of the cell.
Naelea felt sad seeing the chromatid separates. After that, Anaphase recited their farewell and brought Naelea to the next phase, which is the Telophase.
That's the entirety of it, and I hope you know what I'm all about. Bye! Thank you, Anaphase!
Naelea was welcomed by the joyful Telophase.
Hello, hello! I'm Telophase and in the final stage of mitosis.
Oh, you are.
Hi, Telophase!
After the greetings, Telophase started to share information to Naelea.
Telophase begins as anaphase ends. During anaphase, the chromosomes or chromatids on the metaphase plates are separated and dragged towards the opposite poles. And when the chromosomes reach the pole for which they are intended, telophase can begin. In order to allow the new cells to begin producing the necessary proteins and to protect the DNA, a nucleus must reform in each cell.
That's cool!
Telophase then showed Naelea what is happening in her face.
Whoa! So this is how it happens?
Telophase continued.
Yes, Naelea. Did you know that during telophase, the sister chromosomes, one sister chromatid, have you now been segregated to for the two folds of the cell? The mitotic spindle is no longer necessary because the chromosomes completed their journey. The tubulin dimers fall apart. and much of the microtubule network is disassembled.
Naelea suddenly asked, and Telophase answered joyfully.
So, what happens to the spindle and to the tubulin dimers when they broke apart? What happens to the chromatids?
What a good question. Well, you see, during the mitosis, each duplicated chromosomes is evenly divided. Thus, during telophase, two identical nuclei are created. These cells will function in the same way and can be used to build an entire organism from a single zygote or replace cells which have been damaged. Meiosis, on the other hand, produces cells that differ in the genetics they carry.
Okay. Thank you for answering.
It is time for Naelea to head out now, so she and Telophase said goodbye to each other.
I hope you know me well. Bye, Naelea.
Guess I'll be heading out of this microscope now.
Telophase then realized she forgot to tell something, so she spilled it out.
Oh no, you still have to meet someone.
I thought you were the last stage. You told me you were.
Oops, I forgot about cytokinesis. Sorry, go ahead.
Naelea walked a short distance to where Cytokinesis is. Cytokinesis welcomed her as well, but he looked a bit annoyed.
Oh, hi there, do you know me? No, you don't? Nice. Hey, I'm Cytokinesis, the final process in eukaryotic cell division.
Actually, I know who you are. Telophase told me.
Cytokinesis frowned disappeared. He looked happy knowing that Telophase remembered him. His facial expression turned annoyed again when Naelea revealed what really happened earlier.
Finally, that girl always forgot about my existence. There are times where students walk past me and go out of this microscope.
Well, she forgot about you the first time, but she remembered you when I was about to leave.
I know it.
So, since you are the last space, what happens in your base? Does the cell divides completely? I mean, totally, or is there something else going on?
Good guess. I divide into three cytoplasm, organelles, and cellular membrane. Cytokinesis typically occurs at the end of mitosis, after telophase, but we are independent process. I move in the cell caused by the same spindle network that was responsible for the separation of the chromosomes. Parts of the spindle responsible for moving chromosomes break down in late cell division to be used in restructuring the two new cells.
After that explanation, Cytokinesis said goodbye, Naelea as well.
I hope you get to know me well. Bye.
Thank you so much. Arigatou gozaimasu.
Naelea looked up to the lens of the microscope. She looked happy with all the knowledge she gathered to get back at her own size. She has to say the magic.
Magic microscope. I figured out my what, hows, and whys. Please get me back to my normal size.
Naelea returned outside of the microscope. She said goodbye once again to the faces.
I'm back. Oh. Goodbye, friends. See you again next time.
Naelea also thanked Devin, aka me, because I am the greatest invisible being on earth. I'm the best creature ever existed. I am always there in times of need.
Please put an end to this.
Okay, I'm out.
Goodbye, everybody. I am Devin, and I must say, curiosity is a mind in search of knowledge. See you next.
Devin, help me. I need to know about the meiosis.